So, having set up all the background necessary, let us just now remind ourselves of the computational problem and how we can take a deep learning solution to the problem. Okay. This particular solution, deep learning solution, is called DeepBind. This architecture was proposed by researchers at University of Toronto, Brendan Frey and co-workers. So, that is the particular deep learning model that we are going to look at, and we are going to look at the architecture of the model and how that model is trained, both in terms of the parameters and then how the hyperparameters of the model are also calibrated. Okay. So, what we have seen so far is biological question that proteins binds to RNA or DNA sequences in a highly selective fashion, right. They do not like to bind to any random sequence, they are very selective in order to control many cellular processes, right. And then also to these sequences it binds to certain substrings or motifs within these sequences. Okay. So, can we identify or predict which sequences will bind to a protein and which motifs within those sequences will bind to a protein. Okay. And so, the computational problem, we have looked at two different problems, right. One is called as binary classification. Given an input RNA or DNA sequence and given a particular protein of interest, we are going to build a different machine learning model for different proteins. Okay. Let us say we fix a protein P, some transcription factor, let us say. Then we want to predict if this P will bind to the sequence S or no, okay, or 0 or 1, that is the binary classification, two classes that we are going to predict, one of the two classes we are going to predict. Or else it can be a regression problem where we are predicting a continuous valued outcome, where we want to know the strength of the binding or the binding affinity of the protein P to the input sequence. Okay. And what are some challenges here? We want to know whether such a predictive model can be trained from noisy experimental data on protein DNA or protein RNA binding events. Okay. So, there are many data sets available from ChIP-seq, CELEX, PBM, CLIP and other experiments, okay, and then these experiments have different forms of noise, and can we train a reliable model from noisy experimental data sets, okay, and the training set could be, like I said before, positive and negative sequences in terms of binary classification, or a set of sequences and their binding scores in terms of the regression problem. Okay. Once we have built such a predictive model, the most important thing is that in order to gain confidence on the model, for biologists to trust the model, right, for bioinformaticians to trust the model, we need to be able to interpret the model, not just view it as a black box. So, and it is great that DeepBind, this is one of the very first applications of deep learning in bioinformatics. At that time itself they gave careful thought to it, and then they were able to derive these motif PWM matrices or these motif profiles, right, from the learnt model, from the trained model, and they were also able to predict the effects of disease causing mutations on these motifs. Okay. So, that is also a challenge that was at that point, and DeepBind was able to solve these both these challenges. Okay. And this DeepBind paper was by Brendan Frey, the last author is Brendan Frey, and co-workers, researchers at University of Toronto and other affiliated collaborative institutes. And like I said before, it had a uniform treatment of diverse RNA or DNA binding data sets, and then it had a deep learning architecture, and then proper hyperparameter tuning and systematic training procedures. And the post processing was also not just stopping at viewing results out of the predictions from the model, but there was post processing to figure out what are the disease causing mutations, what effect will it have on the motifs, what are the actual motifs that these models internally are learning during the training phase, and so on. Okay. And the primary source of figures or text in this particular set of slides is all from this paper, predicting the sequence specificities of DNA and RNA binding proteins by deep learning. This is from Nature Biotechnology 2015. Okay. And I have already said before, it is a well written paper, the figures are very good illustrations, so I have used them as is in this particular set of slides, okay, the text as well as the figures. So, it is a highly recommended paper to read. So, beyond the slides, if you find it this concept of applying deep learning to bioinformatics intriguing, then I highly recommend you to read both the main text and the supplementary text of this paper. Okay. Okay. So, what is the overview of DeepBind in the author's own illustration? We have these high throughput experiments, protein binding microarray, CELEX, ChIP or CLIP experiments. So, ChIP is for DNA binding proteins, CLIP gives you the ideas of RNA binding proteins, right. And then you have this machine learning model that, because there are like terabases of sequence data that these models would have to analyze, and there are many parameters and hyperparameters in this model that need to be tuned and trained properly, it needs massively parallel compute resources, okay, and that in the form of a GPU. Okay. So, these days GPUs are very common, but then at the time of 2015 it was also common, but then relatively less popular than now. And so, the authors did a lot of fine tuning and coding from scratch. They did not have any libraries that they could just tap into. They just did a lot of coding from scratch to write the algorithms for training and calibrating these deep learning models. Okay. So, using massively parallel compute in the form of GPU servers, they were able to get these large scale terabases of sequence data and then learn different models, right, for each transcription factor of interest or each RNA binding protein of interest, right. And then they were able to get these models one per protein, right, and then from that they were able to figure out what the motifs that are learned during the training phase of these models, and then apply it to understand how the motifs are present near upstream or downstream regions of these genes in order to regulate the genes, and then do these mutation map analysis to figure out the effect of disease causing mutations on these particular motifs, whether it will increase or decrease the binding strength of these motifs, and also to figure out thereby its effect on mutations that are present in individuals, right. This is called as personalized or precision medicine. And one of the interesting things I have already said before is that it was looking at a wide variety of DNA and RNA binding data sets with different noise levels, different technologies done at different centers and so on, and it was a very nice study that unified the analysis of all these diverse data sets into one framework. So, having seen the overview of the deep learning model, this DeepBind, let us actually look into the exact architecture of the model. Okay. This DeepBind model has several stages that successively transforms the input data, so that finally you get a prediction for whether each sequence binds to a protein or not. Okay. So, here is all the input sequences from the training data, right, and then it goes through these different stages: convolution stage, rectification stage, pooling, and then finally the neural network stage. And in the end you will get for each sequence the predicted y, this f of s, right. So, we have seen this before. For sequence Si, right, it is going to do a prediction f of Si, and then you are hoping that that will be closely approximating the yi, which is the ground truth targets. Okay. These outputs are your predictions and the ground truth targets. Okay. So, these sequences are successively transformed through these multiple stages to get these final scores, one score per sequence, okay, or one function evaluation, one prediction per sequence. So, with the hope that f of Si, this f that is what we have learned, is similar to the ground truth value yi. And what are these different steps? These are these convolution steps, rectification steps, pooling steps, and neural network step. And what does each of it do? Now that we have reviewed the concept of a PWM matrix, if you recall, the PWM matrix is a 4 cross M matrix, right. You have this ACGT, and then you have this M length motif. Each column is going to be a set of probabilities that add up to 1, right. If you remove that constraint that each column has to sum to 1, okay, if you remove that constraint, then whatever we used to call as PWM or profile before now gets called as a motif detector. Okay. In a deep learning model, you have quite a bit of flexibility. You do not have to enforce that each column has to sum up to 1, you can just let it be any real value, right. And then automatically, because of this huge number of input sequences and the labels that they have provided on the training data for the sequences, the model should be able to infer what these entries are without having this constraint of summing up these columns to 1. Okay. So, a PWM matrix where you do not have the sum to 1 constraints for each column or the non-negative values for each column, right, it is called as a motif detector matrix or a motif detector simply. Okay. And basically for a particular transcription factor for interest, okay, this model is learned for different transcription factors, different models are learned. So, now we have a particular transcription factor P of interest, right. For that P, the model does not know initially what its PWM matrices are. So, it has multiple candidate matrices in its kind of set of matrices that it is trying out, and those are called these motif detectors. In this particular case, the number of motif detectors is going to be 4. Okay. For this particular transcription factor, it is going to have 4 motif detectors in its library of motif detectors, and those motif detectors, one of them is shown here, right. This is this 4 cross M matrix, right, and this is the logo representation of that matrix. And so, these motif detectors using our standard scanning procedure is scan through each of those sequences. Okay. So, then what do you get for the sequence 1? Let us say you have length n1, sequence 2 is length n2, sequence 3 is length n3, and so on. Okay. And so, because you have 4 motif detectors, you are going to scan each of those motif detector across the first sequence, and you will get the output of this convolution, right, output of computing this probability like operation, right. You are just basically multiplying, you are selecting the right entries in these in this matrix, and then you are just simply multiplying all of them, right. And that operation of the scanning operation is basically a convolution, a 1D convolution. And then so you will get essentially these 4 motif detectors cross roughly n1, okay, close to n1. And so, that is this n1 cross 4, then you have n2 cross 4 here, then you have n3 cross 4 here, and so on. Okay. So, these are the outputs of the motif scans, and you can see here that certain motifs, right. So, this first motif is present in this particular sequence, the last sequence, it is present in this third sequence, it is present in this first sequence, and so on. Okay. These are places where there is a higher likelihood of the motif to be present. Okay. And then there are certain other regions where you have blue colors or white colors, where the motif, that particular motif is not present in that corresponding sequence. Okay. So, now once you have that, we are going to clean up this output of motif scan using this rectification stage. So, rectification we will see more carefully later. Very small values that are close to 0 or less than a particular positive threshold are going to be clamped to 0, and then all the higher positive values are going to be left as it is. This is also known as a ReLU unit in machine learning terminology. So, it is going to rectify, it is going to correct the input, so that small values in magnitude are removed or negative values are removed, and only the positive values above a certain threshold are kept. Okay. And then you have a pooling, where we have already discussed about max or average pooling, right. So, you have now a different signal along the length of a sequence, I need to convert it into one particular value per sequence, right. And so, now you have again the 4 motif detectors, now you have the number of sequences, these are the number of sequences, and so you have one score per sequence per motif detector, right, and that is achieved by basically looking at the overall length and then taking a max or an average after the rectification stage. Okay. Finally, you have this 4 cross number of sequences, I need to get just the number of sequences, and there you use a neural network with certain number of hidden units. So, 32 in this particular paper, and then you have the weights associated with those hidden units, and those are then combined together to get a series of predictions for each sequence. So, if you start with the sequence Si here, right, it is going to give you this f of Si that has been obtained by all of these 4 operations, right, 4 stages, and then we will train this model, we will set these motif detector values, these thresholds that are used for rectification, these weights, all of these are the current model parameters, right. During the training phase, these will be iteratively updated, so that the outputs or the predictions and the targets get closer to each other, okay, that the f of Si, this f which is this overall operation, becomes as close to y as possible. Okay. So, here there are a few numbers that are shown here. In the case of sequences, these are about 14 to 101 nucleotide in length. So, these N1, N2 and N3, all of these are roughly 14 to 101 nucleotides or base pairs in length. It depends on 14 and other smaller numbers are for RNA binding protein data sets, and for DNA binding protein like transcription factor data sets, it is closer to 101. Okay. And then how many parameters in each length M motif detector? We have already seen that this is 4 times M, right, and then M is of the order of 14 to 32 nucleotides. Okay. Again, that depends on the data set. For RNA binding, it will be closer to the smaller number, for DNA binding it will be a little bit larger numbers. Okay. Now, this is DeepBind model architecture in pictures. I hope it gives you an intuition, but then it does not beat seeing it in actual formulas, okay, because that gives you a further understanding of the model, and the model is really these 4 stages written as functions. Okay. So, if you have a sequence S, we want to learn this model F, that is in 4 stages. First, you apply a convolution, and these M are the motif detectors. If there are 4 motif detectors, there are 4 motif detectors times 4 times M, right, for each motif detector's number of parameters, right. So, then using those motif detectors, you do a motif scan over all these sequences, that is the convolution operation. So, that in the deep learning lingo or signal processing lingo, it is called convolution. Then you have a rectification stage, right, where you have these thresholds B that is applied on the output of these motif scans or the output of these convolutions, and finally or last but one step, you have pooling, where you do the max or average pooling to get a value per sequence per motif detector, and then the final network then will combine the values for each of the motif detector across a sequence to give you a single score for a particular sequence. Okay. And let us just see what the ReLU or the rectification is. So, this rectification B of a number, let us say a value V, is going to be V if V is at least B and 0 otherwise. Okay. So, that is what is given here. It clamps all the negative values to 0, right, and then isolates positions with a good pattern matching by shifting the response detector by Bk. Okay. In terms of ReLU terminology, right. So, this ReLU of V minus B is going to be exactly the same as rectification B of V. Okay. So, ReLU is a function that will clamp all negative values to 0, okay, and that is exactly what is done here. Okay. So, the pooling stage, we have already seen that we can do maximum and average pooling to pool the motif scan signals across the length of a sequence into one signal. Okay. We are not going to go into the exact architecture of this nonlinear neural network that is shown here, but suffices to say that it has certain weights W, and then it will take, it is a function, a nonlinear function that is learned, that will map a set of scores, right. If you have k motif detectors, right, k was 4 here, and then it was 6, it will be 16 in default, right, in the paper that they use 16 motif detectors. So, this neural network is a black box, you can think about it, that will take these 16 values or k equal to 16 motif detectors, 4 in the previous example, for the particular sequence S, right. You have these 16 values now, and then it will give you a single value, which is this f of s, okay, which hopefully is close to the true y. Okay. So, that is the model architecture in functions. And how do you actually represent the sequence? So, deep learning model is simply a series of matrix vector or matrix multiplications, right, and then so you need to represent everything in real values. You cannot just have symbols like A, C, G and T. So, for that we are going to use something like a one hot encoding. What does it mean? If you have something like A, T, G, G, and then also there may be nucleotides that are referred to as N, which stands for any of the letters A, C, G, T. You just pad it with N. So, in this particular case N N, A, T, G, G and N N. Okay. You are going to pad it with m minus 1 n's in the beginning and end if you use a motif of length m. Okay. And then you look at all the so that if you do a sliding window, this motif scan, then all the positions are carefully handled. Then you represent this as a n plus 2 into m minus 1 by 4 matrix. Okay. These columns represent A, C, G and T, right. The rows represent the sequence, which is n in length, and then you have m minus 1 over here and another m minus 1 over here. Okay. And you can see here why this is ATGG, right, ATGG, because this corresponds to A, right, and then this corresponds to T, and this corresponds to G, and this corresponds to G. Okay. And then these 0.25 all says that in that first 2 and final 2 positions you have an N. Okay. And again just reminding you that these the sequence length N is of the order of 14 for RNA binding protein sequences and about 101 for transcription factor or DNA binding protein sequences. And similarly, the motif length they tried in this paper different values of motif length ranging from 14 to 32, that is the value of m. Okay. So, now that we have seen the deep learning model architecture both in pictures and in actual function notation, let us just see how to train the model. What does it mean to train the model, right? So, if we look at this notation, right, we have these parameters at every stage, right. So, W is the set of parameters for the neural network stage. Pooling does not have any parameters because it is simply maximum or average of a set of numbers, but this rectification has a threshold parameter B, right. Things that are above B are left as it is, that are below B are made to 0, right, forced or clamped to 0. Convolution layer similarly has these motif detectors. If you have k motif detectors, then you have k cross 4 cross m number of parameters that the model needs to learn. Okay. So, the model will have to start with random values for these parameters, but it will have to eventually converge to a value that will make the predictions, right, f of s close to the true y. Okay. F of si should approximate the true y. Okay. And that is exactly what is done in training. So, you have for a particular sequence all these operations that are done using the current model parameters, right, m over here for the motif detectors, b over here for the thresholds, and w over here for the weights and so on. All of these parameters are just represented in this whole slab of parameters, and in order to do your predictions, right, which is your f, all these parameters, let us say I call it as some capital W of Si, right, and then you want this to be close to the ground truth yi, okay, these ground truth targets yi, okay. And if your initial set of random parameter values that you assign to these different parameters does not have this approximation, then you look at the difference between this prediction f of Si and yi, and that will give you these prediction errors, right, which is some form of distance between f of Si versus yi, right, using your current set of parameters W. I am going to call it as capital W. Okay. And then you can use something called as a backpropagation algorithm that will help you to update the current parameter values and then change the m, change the thresholds B, change the weight W, so that your f of Si is now a bit closer to yi. Okay, and not just for the ith sequence, but across all the sequences. Okay, these errors accumulated across all the training data sequences, you wanted to get it closer to the true value yi. Okay, and that is the whole concept of training. So, it is quite simple in that sense. So, you have certain delta values that you are going to either increase or decrease the parameters, and that is this update step that is shown, so that the new parameters will give you an fw of Si, this fw prime of Si using the new w prime, right, that will get a bit closer to yi than the current set of parameters across all the training sequences. Okay. And then what exactly is it in formulas? Again, we have seen it in pictures, it is good to see it in formulas to see to make sure that we are concrete about what is being done in the training. Okay. So, the training objectives is basically to find the parameters that will minimize the loss function, or in this particular case a penalized loss function, so that you do not come up with very complicated models that will overfit the training data, and overfitting is an issue because then that model will work very nicely for the training data, but it will not generalize to unseen test data. Okay. So, because of that we need a penalized loss function. So, this loss function will tell you how far off yi is from f of Si. Okay. So, yi and f of Si, how different they are. So, that is the loss. Okay. And the penalization will make sure that simple models are learnt, simple models are encouraged. Okay. And so, let us look at the exact form of this training objective function. So, this is also known as the empirical risk or loss. So, that is why it is denoted as R. So, this empirical risk R of W, sometimes also denoted as R hat of W, right, is going to be the average loss incurred across all the training samples. So, there are n training samples, okay, and then you look at f of I, which is what we used to denote it as f of Si, okay, and then you have t of I, which we used to denote it as yi. Okay, I have just changed some notations here. Yi is the same as t of I, which is the target or the ground truth. F of I is the prediction of the neural network, this overall DeepBind model on the ith sequence f of Si. Okay. And so, you look at some form of a loss between how different this f of Si is from yi, you want to minimize this loss, but at the same time you do not want to make the model too complicated, you are going to penalize very complex models. So, this is the penalty term. Okay. So, these are the m is a vector, right. So, and w is a vector of parameters, this w is a vector of other set of parameters for the different stages of the DeepBind model, right. And so, for the parameters that are used in different stages of the DeepBind model, we are saying that their L1 norm should be small. Okay, if these different parameters are then forced to 0, many of these parameters are forced to 0 to minimize this penalty part. If you allow more complex models, then each of these values is going to be very high. Okay. In order to discourage those complex models, we have some weighting beta 1, beta 2 and beta 3. These are hyperparameters that will differentially weigh these 3 different terms, but what it will essentially do is you want to minimize the loss, right, but at the same time you do not want the model to be too complex. So, this and this part are the trade offs. Okay. If you minimize the loss to almost 0, then you will have to make many of the entries in M to be non-zero, many of the entries in your parameters W to be non-zero and many entries in this small w to be non-zero, and that will increase the L1 norm and it will increase the penalty. Okay. But then if you lower the penalty, then the loss may increase. So, then there is a balance that you need to achieve between the first term, which is the loss term, and the second term, which is the penalty term. Okay. And that is why we are trying to come up with a penalized loss function that will discourage overfitting and encourage generalizability of the trained model to perform well in new unseen data sets. Okay. So, now what is exactly this loss function? For regression and classification, you have two different loss functions. For regression, where you are predicting a continuous binding score, it is simply the mean squared error 1 by 2 into f minus t whole squared, okay, the difference between the prediction and the actual value and the squared error between them. For classification problem, you want your prediction, right. You have a prediction where sigma f is going to be your probability, f is going to be some continuous value between minus infinity and plus infinity, and sigma f is the sigmoid function which will convert a value from minus infinity to plus infinity to a value between 0 and 1. Okay. This is going to be the sigma of f that is given by this function 1 over 1 plus e to the power minus f. Okay. So, this is f, this is sigma of f. Okay. So, you have a value that a continuous value f is converted into a probability between 0 and 1, and you want the probability to be high for t equal to 1 sequences. I want sigma of f to be greater than 0.5 for t equal to 0, which means that this sequence does not bind to the particular transcription factor of interest. I want sigma f to be as low as possible, less than 0.5. Okay. And this negative log likelihood is going to capture this relation in a quantitative way. Okay. And this is also known as a cross entropy loss, and it is a standard loss function used for binary classification problems. So, you have this mean squared error loss for regression, and then this cross entropy loss or this negative log likelihood loss for classification problems that you substitute into this loss. And so, you have this first term, this average loss across all the sequences, and then the second term, which is the penalty, and you want to balance these two terms when you are doing the training. Okay. You want to set your training is essentially, if you were to write it as one equation, arg min over all the parameters, your w parameter, your m parameter, your small w parameter and so on, r of w, right. All of this, let us say I call it as a vector w, right. This is the vector w and this is your vector w star. Okay. The training process is going to figure out this optimal set of parameters, this vector w star that comprises all these motif detectors, these neural network and all these parameters on all these stages, and is going to figure out the value of parameter that will minimize the penalized loss. Okay. And that is the whole process of training. So, now actually how do you figure out the optimum value? If w refers to all the parameters you have, this w vector, you want to find out the optimum value of w that will minimize your penalized loss r of w, right. And so, this gradient descent update rule is one particular way to solve this optimization problem for a general function, including this risk function. What it says is that you start with some initial values w naught that you randomly initialize, okay, random initialization of all parameters with some random values. Okay. Then in every iteration you start with the current value w of t, and then you make some little additions or deletion, I mean little increase or decrease to these values. Okay. So, eta is called the learning rate and it is a small value. You choose learning rate to be a very small number, let us say 0.01 or 0.1 or something, and then this is the gradient of the function r, okay, evaluated at the current set of parameter values w of t. Okay. So, a gradient of a function r is basically the set of partial derivatives, right. So, dou r over dou w1, dou r over dou w2 and so on, where all of these are dou r over dou w some value q, let us say, where q is the total set of parameters. Okay. So, you have a vector of all these partial derivatives, you compute your partial derivatives and then evaluate it at w of t, and that according to a basic result in calculus tells you that the gradient at a particular point points in the direction of the maximum descent of the ascent of the function. So, if you take a minus sign, then you are pointing in the direction of the maximum descent. Okay. So, then that is the direction in which you need to take a step in order to reduce the value of the function. And so, that is what exactly this update rule does. It takes the value of w of t and then it decreases it by a certain value, which is given by this eta times the gradient of r evaluated at w of t, right, and then that is taken to be the new value of w t plus 1. Okay. If eta is small enough, then the new value of w t 1 r evaluated at the new value will be smaller than the r evaluated at the current value. Okay. And all of this rests on certain standard facts of calculus that are listed below, but it suffices to say that the particular fact that we are using here is mainly the point 2, that the gradient gives the direction of the maximum rate of change of a function r in the neighborhood of a particular point w. And how do you actually compute the gradient at the current parameter values? You use something called as a backpropagation algorithm. If you take a proper deep learning course, you will get into it in a whole set of details, but we are just using this deep learning solution as an application to a bioinformatics problem. So, we are not going to characterize the solution on how this backpropagation works to compute the gradients exactly here. Suffices to say that these are all the parameters. M are these convolutional stage parameters, B are the thresholds for the rectification stage, this is the neural network parameters and so on, before and after the hidden states, right. All of these parameters are used in a forward sense. So, this is the forward sense when we are doing a prediction, right, and we are taking a sequence and then predicting f of s so that it is close to the true value y, or we call it as t as the target, then we are going in the forward direction. Now, if you want to figure out what is the gradient of r with respect to all the parameters w as this w vector, right, gradient of r, where r of w was this loss function plus some penalty, right. In order to compute the partial derivative, you will have to be going in the reverse direction. Okay. The partial derivative of this loss function would first be expressed in terms of partial derivatives with respect to these w, and then that in terms of the derivatives with respect to this capital W, and then there is a pooling stage has to be specially handled. There is no parameters there, but then in turn you will go back to the parameters b, and the gradient of the loss function of w with respect to b will be calculated, and so you will be going in the reverse direction of how you are doing a prediction, and so that is why it is called a backward direction, and so you are propagating your errors in a backward fashion. So, the errors that you see delta f is going to be essentially f of s minus your true value y, right, and that is going to be propagated down to know how to change your w, how to change your capital W, how to change your small b, and how to change your capital M, which is the motive detectors. Okay. So, let me just make one simple point. The backpropagation may seem like a mysterious algorithm if you have not seen it in deep learning courses, but for those who have never seen it before, it is simply a way to implement the chain rule of differentiation. Okay. Here you have a function, right, f, which is represented as a composition of different functions, right, and let us just take a simple example. So, in this particular case, f is going to be as a function of the parameters, right. So, it takes the input argument s, but let us say that it is a function of the parameter w, right, and then you have let us say two different functions g and h of some w, okay, where these are the parameters. Okay. And we are now trying to view it as a function of the parameters, right, across all the sequences, accumulated across all the sequences, and then we are just trying to do what is dou f over dou w1, let us say, the first component of this vector of parameters w. Now, by the chain rule of differentiation, you know that this dou f over dou w1 is simply going to be dou g over dou h into dou h over dou w1, right. And so, you can see that this is just simply the chain rule. If you have more than two functions, there will be 3 terms over here, and now we have 4 functions here, 1, 2, 3, 4 functions here, and so there will be 4 terms, and then calculating those 4 terms have to happen in this reverse way because of the chain rule of differentiation, and that is exactly what backpropagation does. It backpropagates the error, which is f of s minus y given as delta f, and that backpropagates to the first set of parameters little w, capital W, little b, and finally capital M, which are the motif detectors. Okay. And the final process in training is actually hyperparameter tuning. There are just very many hyperparameters in the model. The number of motif detectors, we said that in the example it was 4, but then the default value used was 16. How did they come up with this value 16? Right? And then there is this learning rate in the gradient descent algorithm. We had this eta, right? So, we said that if eta is small enough, then you will be, the gradient descent algorithm will be able to find the minimum W star that will minimize the risk function, right. So, what value of learning rate eta should you set? So, all of these are different hyperparameters of the model, and one of the challenges in deep learning is to figure out the right set of hyperparameters, and for that we need to do, it was at that point in time this this work was published around 2015. There were a combination of manual approaches, and then there was interest in doing automated calibration or automated hyperparameter tuning approaches, and this paper did a careful automated approach, and that made the performance of the model very good across a diverse set of data sets. Okay. And so, that was one of the good practices that I want to also explain here, because it is useful for not just training and hyperparameter tuning of this deep learning model, but if you are interested in other deep learning models also, you will find it useful. And what is that particular point that I want to refer to? Okay. So, for each data set they divided it into training and testing, and the test data that is shown here is never seen during calibration or training. Okay. That is something that is very obvious. You should never use the test data until in the very final step where you are testing your model, one final model that you have obtained. Okay. But oftentimes when we are coding things, we just keep seeing the test data again and again, and then unfortunately data leaks from the test into the model. Okay. But they did a good practice, they never saw the test data, it was set aside and then used only in the final step. Okay. The rest of the data was used for calibration or training. Calibration, they refer to it as hyperparameter tuning, and then the training is figuring out the values of the hyperparameters, the optimal parameters W star. Okay. Let us say that theta refers to the different hyperparameters. So, like learning rate, and you have degree of momentum, the batch size in terms of the size of the training data that you have, and so on and so forth. Okay. If you have taken a deep learning course, you will understand some of these parameters, hyperparameters, but otherwise also it is okay. You just have learning rate in mind, the number of motif detectors, those are the kinds of parameters that are called hyperparameters. Okay. And each of those hyperparameter can take, let us say, 5 values, right. And if there are 3 hyperparameters, then you have 5 times 5 times 5, right. So, that is 125 total configurations of the hyperparameters. You just randomly sample 30 configurations from that. That is referred to as this theta 1, theta 2 to theta 30. Then for these particular hyperparameters, you train the model on your particular data set, and then you get the best accuracy, which is evaluated in something called the area under the curve, area under the ROC curve. It is like a measure of how well a binary classification model does. Okay. And in this particular case, for regression you will be looking at minimizing the mean squared error, but in terms of binary classification you are trying to improve the classification accuracy or this area under the ROC curve. Higher the area, closer it is to 1, it means the better the classification model is. Okay. Across all these 30 runs, then you look at which value is highest and you select that hyperparameter to further train. Okay. And then because there is a lot of stochasticity in training a model, okay, we saw here in the gradient descent that you start with a random initialization of all parameters W naught, right. And also your R of W is not accumulated across all the sequences, you randomly split your sequences into small batches and then you compute it in those batch steps of batch, that is called as a stochastic gradient descent. Okay. So, there is some randomness and stochasticity in the process. So, even if you feed in the same training data, the parameters W star that are trained will be different when you run it each time with a different random seed. Okay. In order to combat that, they did a 3-fold cross validation. They divided the training data into these 3 parts and they used 2 folds for training and 1 for validation. So, because it is 3-fold, then you will have 3 such validation data sets, and so across these 3 runs, then you compute the average accuracy, and then that average accuracy is reported here in this final table, and then the whatever hyperparameter configuration, whichever of these 30 had the highest average accuracy across these 3 folds, right, 3-fold cross validation, that was selected further. Okay. Again in the training phase, now that you have selected the hyperparameter, right, now you are going to train, you do not need to just stick with training only 2 folds, I want to use all 3 folds to train. Okay. Again I have this stochasticity problem, every time I train, because of the gradient descent it will give you different parameter values. I just try 3 attempts or 6 attempts, right. In this case it is shown as 3 attempts, in the paper they actually did 6 attempts, right, to train, to use all the training data to train for this particular hyperparameter. So, here theta 2, the second configuration of hyperparameters is chosen, and all 3 are now used to train the parameters W star. So, W star first run, W star second run, and W star third run, right, using all of the 3 folds of the training data, the same training data, right, instead of splitting into 3 folds, we are going to use all of the training data. And then now you do it across all of them and you take the best one. Okay. In this particular case it is 0.97. So, the best among all these training candidates is now used and find as the final model to apply once to the test data, right. All these parameters M, B and W, the motif detectors, the rectification thresholds and the neural network parameters, all of that are used to predict for the test data what the labels are, right, in terms of binary classification, whether the sequence in the test data, the test sequence binds or does not bind to the protein of interest, and in terms of regression model, what is the strength of binding. Okay. Using that in the classification model, you can compute the test accuracy reported as an area under the curve, the receiver operating characteristics or ROC curve. Okay. And that gives you a 0.93 in this particular case. Okay. So, this is a very good practice for how to tune hyperparameters and how to combat some of the randomness or stochasticity in the training process. Okay. So, I hope the kind of amount of time I have spent on this is mainly to see that if you can use this in your other deep learning projects as well, whether it is in bioinformatics or in other application domains.